Hello students, welcome to EPG Pathshala. I am Dr. Hari Prasad, Associate Professor at the Department of Biophysics, All India Institute of Medical Sciences, New Delhi. Today we are going to talk on the module Structure Prediction Methods using ab initio and threading under the paper Bioinformatics. After completing this particular module, you should be able to understand Introduction and Principles in ab initio modeling, protocols used to create ab initio models, softwares for ab initio, advantages and disadvantages in ab initio modeling, introduction and principles to modeling by threading, protocols used to create models by threading, softwares that are used for threading, advantages and disadvantages in threading modeling, and finally conclusions. Protein structure prediction is very important and there are certain methodologies that are used: homology modeling, ab initio, and threading. I will now be talking to you about aspects regarding ab initio. Ab initio means from the beginning. The method predicts the structure of a protein from the scratch using sequence information by applying Newtonian force to achieve a thermodynamic stable native-like state. It is very useful in situations where the query sequences do not have any suitable template in the protein structure library for either homology modeling or threading. We also apply what we call as a Anfinsen's theory. Anfinsen's theory states that protein native structure corresponds to the state with the lowest free energy of the protein solvent system, and that this particular information is contained in the amino acid sequence. The basic idea is to build empirical function that stimulates real physical forces and potentials of possible chemical contacts within the protein. The methodology of ab initio is very dependent on Anfinsen's theory. Anfinsen's theory states that all the information of the protein folding is contained in the sequence itself, and therefore cell machinery is immaterial in protein folding. This holds true for most of the small globular proteins. Protein native structure corresponds to a state with the lowest free energy of the protein solvent system. The protein energy landscape is very uneven. It consists of many local minima in which partially folded proteins with relatively higher energy are seen. The global minima, representing the native structure, is found deep down the energy funnel, passing through several molten globule states, which is a number of intermediate conformational states between the unfolded states and the complete native state of the globular protein that are located. Thereby, you see the protein sequence at the very top, intermediates in between, which are called the molten globule state, and the well folded protein or the global minima, which is very close to the native structure. Having now understood that the primary sequence of the proteins are just not amino acids that are strung together peptide bond but hold vital information on the final protein structure, software programs are so designed to use certain basic principles and concepts of physics to predict the three-dimensional structure of proteins. Protocols used in ab initio. The very few basic steps that are used in ab initio are: first, the protein representation that assigns rules and restrictions that are to be applied. Then, you have the energy functions that define the total potential energy of all atoms and which can be derived either by physics-based or knowledge-based. We then do a conformational search to identify the global minimum energy state, and this particular method can either use energy minimization, Monte Carlo, simulated annealing, molecular dynamics, or genetic algorithm. And finally, we do the model selection to choose the best of the native-like structures from a pool of decoy structures. Protein representation. Approaches to structure prediction differ with the assumptions they make. It can therefore be represented as three-dimensional coordinates in space or a set of dihedral angle pairs. Softwares differ in the way proteins are represented. The torsion angles are restricted to a finite set of values. Hydrogen atoms are given importance in polar residues. Restrictions are laid for nonpolar hydrogen atoms. Only a finite set of dihedral angles are considered, and bulky side chains are replaced by single pseudo atoms. Energy function. An potential energy function specifies the total potential energy of a system of all atoms as a function of their coordinates. These energy functions are basically a description of a set of functional forms and parameters used to compute the potential energy of the system of interest. These potentials are used as energy functions in the assessment of an ensemble of structural models generated during the modeling. The main parameters include bond stretching, angle bending, dihedral torsion, H bonding, van der Waals interactions, and electrostatic interactions. Physics-based energy functions. Here, the interactions between atoms are based on quantum mechanics and the Coulomb potential, with only a few fundamental parameters such as electron charge and the Planck constant that pertain to quantum mechanics. There is a compromised force field with a large number of selected atom types. Well-known examples of such all-atom physics-based force fields include amber, charm, OPLS, and Gromo 96. You also have what you call as the solvation energy, which reflects the implicit solvation model, the generalized bond GB model, surface area based model, and the explicit solvation model TIP3P. Energy potentials contain terms associated with bond lengths, angles, torsion angles, van der Waals, and electrostatic interactions. Knowledge-based energy functions refers to the static energy potentials containing the empirical energy factors that recognized from analysis of already reported protein structures in the protein data bank. They involve less computational cost. Some of the most famous or well-known softwares that use this particular approach is the ITESR. This methodology could be either be sequence independent, which deals with atomic interaction potential, hydrogen bond potential, torsion angle potential, solvation potential, or it could be sequence dependent where pairwise residue contact potentials, distance-dependent atomic contact potentials, and secondary structure propensities are made use of. Conformational search methods. The protein energy landscape is generally rocky as shown in the figure. It contains many local energy minima and maxima along with intermediate saddle points. But then it finally has only one global minima. The saddle points correspond to the transition states, the barriers that all the molecules must cross if they are to fold to the native state. This is the very point where the slope of the orthogonal function space becomes zero. In order to find a valid ab initio structure model of a protein, it is important to have powerful conformational search method which for a given energy function can accurately and efficiently find the global minimum energy structure. The different methods one could possibly use are energy minimization, Monte Carlo, molecular dynamics, genetic algorithm, and simulated annealing. Conformational search method using energy minimization. Energy minimization is a molecular or a quantum mechanical process to find the lowest energy conformation of a protein. Given a state A with an energy UA, the system makes several changes in the atom position to arrive at a new state A dash with the energy U dash A and calculates the energy in every position. So the process is repeated many times until a configuration with an overall minimum energy is attained as shown in the figure. The different methods in this particular step could be the first order minimization that includes steepest descent, conjugate gradient. Second, the second derivative methods which includes Newton-Raphson method, or one could also use the quasi-Newton methods such as LDFGS. Conformational search methods using Monte Carlo simulations. Monte Carlo minimization is an important optimization and sampling technique that does not use forces but rather compares energies. It extensively makes use of Boltzmann probabilities. Here, one performs the Monte Carlo moves between the local energy minima after local energy minimization of each perturbed protein structure. This can be done starting with a given local energy minima structure A. A trial structure B is produced by random perturbation of the original structure A. It is subsequently subject to local energy minimizations. The acceptance or the rejection of this particular move is based on the metropolis criteria. The multi-start approach takes advantage of the fragment library with multiple random trajectories simultaneously to save the time cost of the procedure. The Monte Carlo method of conformational search uses the accept and reject theory. The fundamentals include making a random move to produce a new conformation R dash. We then calculate the energy change in terms of delta E for the new conformation. Using the metropolis criteria, this particular move is either accepted or rejected and incorporates the Boltzmann equation. If the difference is less than 0, we then take the new conformation and then start the iterative process once again. In this particular way, at every step, a new conformation is either accepted or rejected, and if so, it goes back to the original starting point. A software that uses this particular application is the Linus. Conformational search method using molecular dynamics. The molecular dynamics simulation is aimed to understand the properties of the assemblies of molecules and the atomic level interactions between them. The molecular dynamics simulation solves Newton's equations of motion at each step of atom movement, which is probably the most reliable method, where the force is the product of the mass and acceleration. Long simulation time is one of the major issues of this method, since the incremental timescale is usually in the order of femtoseconds. When a low resolution model is available, MD simulations are often carried out for structure refinement since the conformational changes are assumed to be small. The conformational search method using molecular dynamics simulates the movements of all the particles in the molecular system by iteratively solving the Newton's equations of motion. The most commonly used softwares would be the CHARM and NAMD. To obtain the movement trajectory of atom, numerical algorithms are used for integrating the equations of motions, which are the Verlet algorithm and the leapfrog algorithm. A demonstration of how the protein movement folds is demonstrated in this particular video. The advantages of this particular method are that it is deterministic and provides details of the folding process. The disadvantages, however, are that protein folding reactions usually take place at microsecond level, which is at the limit of the accessible simulation times. Molecular dynamics in terms of principle of movements. For an atom, Newton's equation of motion is given by the formula force is equal to mass into acceleration. The acceleration on this particular atom at time t can be given with respect to position r by this particular formula. The function of time can also be expressed as a gradient of potential energy. Therefore, we can now equate the two equations and arrive at the Newton's equation of motion that can relate to the derivative of potential energy to the changes in position as a function of time. Molecular dynamics involves timescales with the entire mechanistic process of protein folding. It is on a scale from femtosecond to a second. The various possible events that occur during this particular process are bond stretching, elastic vibrations of proteins, the alpha helix folding, beta hairpin folding, and finally the folding of the elements of the secondary structure to form a well folded three-dimensional structure. Conformational search method using genetic algorithm. The presence of large energy barriers and the rocky energy landscape is more likely to get caught in local minima. The genetic algorithm addresses this problem in protein folding more effectively. Unlike the folding of a protein in the Monte Carlo, where typically a single amino acid is changed, a simultaneous change of several angles are carried out in genetic algorithm. This kind of simultaneous change is provided naturally by a crossover operator that is used. The basic operations include the mathematical representation of a set of pair of phi-psi angles for all the residues in the main chain. Each pair of phi-psi angle represents a Cartesian coordinates for a location of the C alpha atoms, and the dihedral angle representation of protein conformations can be used directly to describe possible solutions. We then use genetic operations just like as used in the genetic concept of replication that is implemented by adopting a solution from one generation to the next. And with every passing change, the native structure is thought to be obtained, which is better than the previous generation. In this particular way, mutation operator incorporates changes to the conformation, and the final crossover operation is implemented wherein some approach restrict the dihedral angles as real numbers, and finally the offspring structure will contain a part of each of its parents' structures and will be more closer to the native structure. The minimization and fitness evaluation is then done for this particular model just as described or explained for other methods. One of the conformational search methods is simulated annealing. Simulated annealing is probably the most popular and the straightforward conformational search method. In the energy landscape, when the energy of the system under investigation is rocky due to the presence of numerous energy barriers, Monte Carlo simulations are prone to get stuck in metastable states. The underlying idea here is to accelerate the transition between states separated by energy barriers by modifying the transition probability so that the final energy distribution of the sampling becomes more or less flat rather than bell-shaped, hence apparently decreasing the energy barrier. Adjustment in temperature apparently leads to the flattening effect of energy contour. Simulated annealing as a method of conformational search. As it has already been described, the energy landscape is the mapping of all possible conformations and contains many energy barriers which may easily trap Monte Carlo simulation procedures in metastable states. To address this particular problem, the simulated annealing is incorporated within the Monte Carlo method. Simulated annealing is probably the most popular and straightforward conformational search method. The underlying principle or the theory is to accelerate the transition between states separated by energy barriers by modifying the transition probability so that the final energy distribution of sampling becomes more or less flat rather than the bell shape that we are familiar with, hence apparently decreasing the energy barrier. Adjustment in the temperature apparently leads to flattening effect of the energy contour and is well incorporated in the principles for the probability distribution using the Boltzmann equation. At low temperature, the particular ratio of the delta energy with temperature tends to be equal to 1, therefore it becomes 0 and the move is rejected. At a higher temperature, it becomes equal to 1 and hence the move is selected. So, in actuality, it is like moving all around the energy surface as if the energy surface is squeezed to a flat contour. This allows larger steps that consequently search faster in the conformational space, and thereby at high temperature the energy barrier problem is overcome. Model selection from a pool of decoy structures. Irrespective of the conformational search method used, we arrive at a number of possible decoy structures and then need to choose the final one. The methods that could possibly be used are based on energy or clustering. In the energy-based method, specific potentials and identifies low energy states, for example, as the one that is used in the software Astra Fold. In the other method, clustering, the cluster center conformations represents the largest cluster and is considered as the one that is closest to the native structure. And the software that best uses this particular principle is the Rosetta. The spiker is based on more than 1,000 representative benchmark proteins, each with up to more than 280,000 structured decoys. The best of the top 5 models was ranked as the top 1.4% among all decoys. For the 78% of all these proteins, the root mean square deviation difference between the best of the top 5 models and the most native-like decoy structure was less than 1 angstrom, which reflects the suitability and the adaptability of choosing the best of the decoy structures. Loop prediction by ab initio method. Loops are secondary structural elements that are flexible and connect rigid structural elements like helices and strands. This particular aspect of predicting the loop conformation is very challenging using any method of protein structure prediction. It is important to bear in mind that loops are very integral for protein folding and protein function. They exhibit a greater structural variability than helices and strands. Therefore, the analysis and arriving at the right conformation of the loop is often important but a limiting factor on fold recognition methods. There are a number of software links for protein structure prediction by ab initio. They are the Robetta, Protinfo, Scratch, AstraFold, Rocky, Bhagirath, Charm, and NAMD. These particular softwares are available on the website and be used by one and all. Having understood ab initio, we now will look into the various advantages and disadvantages of this particular method ab initio. First, the advantages. It is deterministic and it provides details on possible aspects of protein folding. Disadvantages. Molecular dynamic methods are very slow. This particular method of ab initio is feasible only for very small proteins that have less than 100 residues. Force fields are not realistic enough. The number of possible conformations are too big and there is a possibility of an exhaustive search. Energy minimization can pose problems. The real structure may not be identical to the model. With this, we come to the end of ab initio. We now move on to another method that is called as threading, that is used for protein structure prediction. So, when I start to introduce threading, I would first like to say that threading was first coined by Jones, Taylor, and Thornier. It is a sequence structure alignment approach used for proteins which do not have structural homologs. It matches a protein sequence to a fold library by using a so-called threading algorithm that assigns each amino acid to a position on the three-dimensional structure for a particular fold class, which is available in the protein data bank. Energy minimization or molecular dynamics is then used for structural refinement and selection of the best model. The protein data bank is an interesting story of unlimited sequence structures with limited folds. To explain, I have two graphs, one on the left and one on the right. The one on the left talks about the growth of the protein data bank entries. Right from the time the X-ray crystallographic analysis was done of myoglobin, there has been a continuous submission of protein structure coordinates, and over the last couple of decades, there has been a steep increase in the structures and has come to finally reach a staggering number of more than a lakh and 17,000. But at the same time, if you see the unique folds of a protein can have, we see that there has been a plateau in the last couple of decades and is not truly proportional to the number of structural coordinates that are being submitted. For the table on the left, we show that in the year 2010, for the 70,000 structures, there existed 1,393 folds. In the year 2015, for the 1,14,000 and odd structures, there was the same number of folds of 1,393. And as recently as 2016, there has been 1,17,000 or more structures with the same number of folds. This means to say that with the increase in the protein structures, there have been structural analysis which relate or show a fold that has previously been analyzed or deciphered. In fact, as shown in the table on the right, the scope classes has clearly demonstrated a particular set of folds a protein can adopt. It could be an all alpha, an all beta, alpha or a beta, alpha with the beta, multi-domain, membrane and cell surface, small proteins, and finally miscellaneous other folds, which constitutes the 1,393 possible folds that a protein can adopt. Principles of threading or fold recognition. Proteins with less than 10% sequence similarity were found to have analogues when aligned structurally. Remote relation can be better detected through structural homology instead of sequence homology. A number of unique domains and folds have been the same over the last few years. Any new protein sequence must therefore adopt one of the well-known folds. There have been very few representative protein folds or domains that have been characterized so far over the last few years. Analogous folds share same structure but have insufficient evidence for an evolutionary relationship. The methodology that is used in threading or fold recognition. As discussed, for a particular query sequence, there can exist only a definitive number of folds that are available in databases such as the protein data bank, the scope, or the CAB. What is done is the sequence is aligned to these structural folds using a scoring function. Out of the possible models that could possibly reflect the low energy states, the model prediction and model optimization finally chooses the best model that could be closest to the native structure or the protein in vivo. Designing of a scoring function. Scoring calculates the most accurate fit between the query sequence and the template structure. It considers the environmental and biological parameters like the environmental fitness potential along with gap penalties. Pairwise interaction potentials between atom pairs of the target sequence and library folds are considered. The potential for any residue is obtained by summing over all the pairwise potentials involving it. Now we come to alignment. Once the library and scoring function is defined, the target sequence is aligned to each library entry. Energy function is used to evaluate the fit between the target sequence and the library entries and to determine the best possible templates. Apart from the dynamic programming, combinatorial optimization approaches such as simulated annealing conditions, random fields, and branch and bound are used to find the heuristic alignment solution. Alignment based on position independent 3D profile method. Here, The residue position in a structure is independent of all the other residues. The query sequence is therefore aligned to physical environmental descriptors on template structural folds. Therefore, these residues in experimentally solved structure library, there is consideration of parameters like solvent access, which could be either buried or exposed, the hydrophobicity, which could either be polar or non-polar, and finally the secondary structure, which could be a possible alpha helix, beta sheet, or a coil, giving rise to a total of 12 possible states. With an increase in such physiochemical parameters, there is an exponential increase in possible conformational states. Alignment based on position-dependent three-dimensional profile method. This method overcomes the major disadvantage in three-dimensional profile method that assumes independence between all positions in the structure. It makes use of position-dependent parameters like contact potentials along with the physical environment and chemical descriptors while performing sequence structure alignments. Most threading methods use this kind of approach to find the best template. Further course of action like loop modeling, sidechain generation, and model optimization is much similar as practiced or explained in homology modeling. Sidechain construction, model optimization, and prediction. The construction of the sidechains is done using rotamer libraries. The score is determined considering the probability of a rotamer and its fitness in a specific position. Scores of all the rotamers are compared for a given amino acid to determine the preferred rotamer to be adopted. Model optimization to bring the structure closer to the real or in vivo conformation is done as by molecular dynamic simulation of the model. Simulation follows the motions of the proteins on a femtosecond timescale and mimics the true folding process. The process ends with a true structural model having minimum energy configuration. Now, having come to the end of this particular topic threading, we will then look into the advantages and disadvantages of threading used in protein structure prediction. Advantages. It's moderately successful. It's good for proteins with less than 100 residues. Disadvantages. The methodology assumes that the current structural library consists of all the possible conformations that could possibly be deciphered experimentally. Less than 30% of the predicted first hits are true remote homologs. The problem of identifying the best alignment is very challenging. High computational cost is involved to screen a library of thousands of possible folds. Energy functions are simplified for efficient calculations and results are therefore compromised. There are a number of software links that are available for protein structure prediction by threading. Few of these are listed here. They are the Phyre, the Raptor X, Prospect, NovaFold, HHPred, and iTessar. The web links to these softwares have also been listed and available for any student to use. So students, now that we have come to the end of this particular module, let us now summarize what we have learnt. Two methods, ab initio and threading, which is also called as fold recognition, are moderately successful methods to predict protein structures. Ab initio requires a sequence, whereas the methodology threading requires a sequence and also a fold library. Ab initio is based on the fact that structural information is contained in the sequence, while threading relies on the fact that all proteins must have a fold that matches the structures that have been experimentally determined so far. Both these methods are based on energy functions and requires algorithms to choose the best native-like model from a set of decoys. Sophistication and high costs are the price one has to bear to get the best of these two protein prediction methods. Thank you so much.